Good day, dear colleagues. I'm about to present to you the results of our work related to information extraction from texts, antiviral agents that are active against
Yes.
viruses.
Excuse me, could you make full screen for your presentation?
I did. 1 second. Is the full screen now?
Oh, probably.
May I continue?
If there are no other choice, please continue this way. Otherwise, we won't have time. Okay.
Viral agents cause many diseases. Moreover, not only the main illnesses are significant, but also accompanying ones. Most therapeutic compounds target key proteins in life cycle of microorganism. HIV infection leads to interaction with many organism systems, especially immune system. HIV infection pathogenesis involves many proteins, some of which are widely known, and some are only about to be discovered. Human proteins that are accountable for containment or spread.
Sorry, but we don't see your slides, just the title.
Now?
Yes. Yes.
Well, human proteins that are accountable for containment or spread of infection through body are called host dependency or host restriction factors. These factors can be considered as targets for anti-HIV drug development. The main source of HIV host interactions mechanisms and potent treatment are texts of scientific publication. Since HIV is known for almost 50 years and claims the lives of almost 1 million people annually, a large amount of texts has been accumulating since. A query to PubMed containing HIV returns over 300,000 results, and the analysis of such a large amount of unstructured text can take the whole life of a single scientist. Therefore, our task was to develop an integral approach that allows us to identify compounds that are active against proteins of virus host interactions using HIV infection as an example. To do this, we have consistently fulfilled several steps. As a first step, we developed proteins and chemical name identity recognition algorithm that allows to detect protein and chemical names within the text. Then, using this algorithm, we extracted information on HIV host interactions mechanisms based on a rule-based approach. As a last step, we performed the identification of chemical compounds that target proteins revealed at the previous stage. Firstly, we have developed a protein and chemical name identity recognition algorithm. Since our algorithm is based on machine learning methods, training sets or specifically annotated corpora are required. To this end, we used Chemintner and drug-prot corpora that include protein and chemical names annotations. Texts of annotated corpora were tokenized in order to present them into machine-readable form. We have chosen optimal ways of tokenization to achieve high accuracy of recognition for both groups of named entities. Received tokens were then labeled according to SOBI marks in order to determine their belonging to the named entities. We have developed a set of features to describe tokens. The full list of them is presented in the table. Since all tokens are semantically related to each other in the context, we also used the features of the neighbor tokens to describe the current. The implementation of the algorithm is a set of scripts written in programming language Python. Recognition accuracy was evaluated according to the common metrics in 2 experiments. The first experiment is a 5-fold cross-validation on annotated corpora, and the second is an evaluation of an external test set, which included manual verification of the results. The value of metrics obtained in both experiments for the tasks of chemical and protein named entity recognition are presented on the slide. Our results are comparable to ones published in related literature. For identification of proteins and genes that involved in HIV host interaction, we collected a set of abstracts from PubMed. Relevance of text was determined using mesh terms. This text was then split into sentences, and the received sentences were sent to the input of protein named entity recognition algorithm. We considered only sentences that contain at least 2 protein names or more, according to recognition results. Sentences of 100 abstracts were analyzed to identify special phrases that indicate interaction. Based on the commonly used context, rules were formed that take into account the occurrence of name and the template phrase and the direction of interaction. In summary, we have created more than 200 rules to extract association between recognized proteins. Sorry. In the end, nearly 3,000 unique interactions within the abstracts were extracted from the text. In order to unify names of proteins, we extracted UniProt IDs using queries to aforementioned database. Also, we retrieved belonging of proteins to organisms. This allowed us to get rid of extracted associations that are not relevant to HIV host interaction mechanisms. In total, more than 700 unique proteins and genes were involved in such interaction. We performed a graphical representation of the result of the step using third-party software. On this slide, you can see a fragment of the network of interactions between HIV and human proteins. Each hub of this graph represents HIV or human protein. It is not surprising that the most represented proteins with the widest network of interactions were HIV proteins, in particular GAG-POL protein, and key human cell receptors, for example, CD4. Results of text mining approach were verified in prospective clinical study. For this, 10 HIV positive subjects were enrolled. They were divided into groups according to the duration of HIV infection, up to a year or more than 1 year. An analysis of gene expression level was made in these 2 groups for the purpose of comparison. In total, 606 differentially expressed genes were revealed. They indicate changes in human organism with HIV infection. Some of them overlap with the results of text mining approach. For interactions, there is literature data on involvement in the pathogenesis of HIV. For those not accompanied by literature evidence, there is an assumption based on the pathogenesis of other viruses that affect the human immune system. We identified several chemical compounds that target revealed in this work proteins. Examples are presented on the slide. Some of them affect human proteins involved in the pathogenesis of the disease, some on HIV proteins. And some of them have already been approved by FDA for the treatment of HIV, and some of them are still in clinical trials. Thank you for your attention. I'm sorry for these technical issues.